In this talk, I will talk about imaging molecular and neurotransmitter dynamics from simultaneous PET and fMRI. And I have no disclosures. The brain consists of networks that are present at various scales. Functional networks span the whole brain level, which can be visualized with functional MRI to investigate underlying neuronal networks. PET, or positron emission tomography, can be used to inform us about brain chemistry at the synaptic or protein level. And in today's talk, I will focus on how various types of stimulation may affect molecular and functional signals dynamically. And the main focus of this talk will be about the temporal integration across modalities. Each modality has its strengths and limitations, and we know that EEG and fMRI, which are covered in other talks in the Symposium, have the ability to track faster events compared to PET imaging. But especially when you do multimodal imaging, it's important to think about all timescales that are important for the intervention that you're trying to study. Neuroreceptor PET imaging is the gold standard for determining in vivo drug binding to a specific target. In this figure, you can see parametric maps of a dopamine D2, D3 receptor-specific radiotracer during the administration of different doses of an antipsychotic drug. And as the dose increases, occupancy measured by PET also increases. Functional MRI techniques allow us to image the brain's functional response or activation due to a stimulus. And in pharmacological MRI, the stimulus is purely a drug, and we can also look at its functional signaling, such as in this case of an amphetamine dose. The simultaneous acquisition of PET and fMRI allows us to connect neurovascular responses to receptor occupancy. And that's a key in the following studies that I will show you, where we combine receptor-specific PET images with fMRI in order to understand this chain of response. And there are two main mechanisms that we look at. One of them is to look at the effects of exogenous drugs, and the other is to try to understand endogenous neurotransmitter release. And of course, each of these chain of events can be measured by various types of imaging contrasts and modalities as well. But specifically, what we use is multimodal imaging with simultaneous PET and fMRI, which I will focus on in this talk. And this table more specifically outlines complementary features of PET and fMRI. And here I talk about specifically neuroreceptor PET and functional MR imaging. Of course, PET is known for its molecular sensitivity to its target, and it can track pharmacokinetics and dynamics, as well as quantify receptor occupancy, all at tracer level doses. The observable dynamic changes are radiotracer dependent, and so that's really key to trying to design your studies. Of course, compared to functional MRI has a better temporal resolution and can observe dynamic changes at a relatively high resolution. But detectable signal changes from baseline are also steady and acquisition dependent, and in most cases, the measurements are not quantitative as opposed to PET measurements. But again, each of these modalities bring their own strengths. When doing combined PET and fMRI experiments, it's really important to consider experimental design, and especially when it comes to dynamic signal acquisitions. There are various ways to do interventions when it comes to PET imaging. A traditional way to do a challenge is to use a bolus injection with a co-administration of other drug doses, or a two bolus design. But there are also bolus plus infusion or within scan challenge of drug doses, which allow you to image signals more dynamically. And of course, in contrast to dynamic fMRI signal changes, PET images are traditionally being acquired on a steady state level over longer periods of time, and so this really needs to be reconciled together with fMRI dynamics when doing experiments in both modalities. So here's an outline of my talk. In the first part, I will talk about pharmacological PET and fMRI, and talk about how dopamine receptor dynamics can be imaged and how we can use PET and fMRI to image receptor internalization, recycling. And then in the second part, I will focus on molecular neurotransmitter dynamics. I will talk about models for estimating dopamine transients and talk about how fast we can really image using PET and other modalities. So just to start off with a little bit of receptor theory of the dopamine receptor here. In a classical occupancy model, we think of the postsynaptic membrane as being occupied by dopamine receptors. In this case, this would be D2 receptors if we are imaging with a radiotracer C11 raclopride. Some of those receptors are occupied by baseline dopamine or endogenous dopamine. And when there's a drug that washes in or endogenous dopamine that binds to these receptors, we would expect there to be a downstream response that elicits an fMRI signal. And at the same time, a PET signal that's specific to those receptors would decrease because occupancy increases. But we also know that this classical occupancy model is not a complete model, but that receptors are dynamic entities on the membrane. And so with certain drugs, especially agonists, as well as endogenous dopamine, when they bind to the receptors, receptors can desensitize, which would diminish a functional response, and receptors can internalize, which then would also decrease the PET signal. And with PET alone, you cannot distinguish between these two scenarios, but together with fMRI and their dynamics, we can try to infer some more a better understanding about internalization. And so that's what we did in these set of studies. We looked at an antagonist, a D2-D3 antagonist, raclopride, and a D2-D3 agonist, quinpirole, with the expectation that the antagonist does not induce receptor internalization, whereas the agonist does, as has been shown in vitro studies. And so overall, we show that the antagonist has occupancy, but shows a positive CBV response localized to the striatum, whereas the agonist shows the opposite sign. But the more important piece of, or the more interesting piece of this data is the dynamics. And we find that with the antagonist, there are similar temporal responses between PET and fMRI signals, which are concordant with a classical occupancy model, whereas we find diverging temporal responses between PET and fMRI that indicate receptor internalization mechanisms. We were really interested in understanding the dynamics of internalization of varying types of dopamine receptors, in our case D1 and D2 receptors. It is known that amphetamine-type stimulants acutely increase extracellular dopamine levels, and that induces internalization. In vitro evidence shows that D2 receptor internalization occurs very fast, within minutes, and that they stay internalized for a very long time, more than an hour and up to 24 hours, really. It has been shown that D1 receptors can quickly recycle back to the membrane first, but again, only in vitro. And so the only in vivo evidence we have is from PET studies where it has been shown that D2 receptor availability stays depressed for up to 48 hours due to receptor internalization. So we designed a study to determine dopamine receptor adaptations over time in order to understand differential recovery times for D1 versus D2 receptors. And so this study was done in non-human primates, and we use a simultaneous PET MRI scanner to look at an initial amphetamine injection at 0 hours, and then we give a second amphetamine injection at either 3 or 24 hours. And then as a second part of the experiment, we also gave a D1 receptor blocker in order to look at specificity of D1 versus D2 signals. Our results of amphetamine effects over 24 hours show that dopamine release occurs with each amphetamine challenge at 0, 3, and 24 hours in the striatum. Our cerebral blood volume data shows that there's a negative CBV change at 0 hours, but there's a positive CBV change at 3 hours, so the sign is completely flipped. And we get a mixed negative and positive CBV component at 24 hours. If we look at the dynamic time courses here, we can see that dopamine receptor occupancy remains elevated at 3 hours, but returns to baseline levels after 24 hours. and the CVV response is markedly different between 0 and 3 hours. If we then give a D1 receptor blocker, we can see that dopamine release is slightly attenuated, but most importantly, we find that the CVV response at 3 hours was effectively blocked, suggesting that this is really D1 driven at 3 hours. And so when looking again at the maps, we see that under D1 receptor blocking conditions, we then eliminate the positive component of the cerebral blood volume response at the 3 hour mark as well as the 24-hour mark, suggesting that D1 and D2 receptors have differential recovery times, with D1 receptors recovering much faster compared to D2 receptors. So this brings me to the second part of my talk, where we'll cover molecular and neurotransmitter dynamics. We were interested in imaging fast dopamine transients, and so we looked at a model of deep brain stimulation in the VTA. And this is a target that is known for its role in reinforcement learning and motivation. It's involved in arousal, and it has been shown that VTA stimulation leads to dopamine release. It's not known what the receptor contributions are, what the whole brain circuit effects are, and what dopamine dynamics look like over longer periods of time. And so we used a non-human primate model where we implanted a microstimulation electrode into the VTA and then used simultaneous PET and fMRI to look at acute stimulation during scanning sessions. Even at the single session level at the fMRI, we can see robust signal changes due to the stimulation, in this case at 16-second separation for 10 minutes. We see changes along the dopaminergic pathways. Now, the simultaneously acquired PET data suggests that there are also activation periods at the single session voxel-wise analysis. And this is true for longer periods of stimulation of 30 minutes or shorter ones where we have a 10-minute on-off block design. At the group level analysis, we again find robust CBB activation in the ipsilateral ventral striatum encoded due to the microstimulation. When it comes to determining dopamine release with C11 raclopride, we found that a new model that required a forward model implementation of a full reference tissue model really was able to capture robust changes that are localized to small areas and allowed a faster converging for estimating binding potential changes, compared to standard models like the MRTM2, which sometimes show an apparent increase in low binding regions. So overall, I'd like to remind everyone that PET is highly sensitive to molecular processes, but the test-retest variability can be on the order of 5 to 10%, and that we need to think about effect sizes, and they need to be large over a sustained period sometimes, depending on how you're measuring your signal. But in that context, and there have been other models like neurotransmitter PET that try to capture time-varying dopamine binding, and simulations have shown that dopamine transients of varying lengths can really be shown on PET time activity curves, but when there is noise in those time activity curves, those decreases become less apparent and are not always visually discernible. And so a novel neurotransmitter PET kinetic model has been proposed to estimate the temporal characteristics of a transient neurotransmitter component in PET data, as shown here. And some of the things that can be done with this model are to look at dopamine dynamics in this example of cigarette smoking, and it has been shown that there can be sex differences where peak dopamine levels in response to smoking is higher in men versus women, and that women responded with a faster dopamine surge in other areas of the putamen. But when using these models, we also introduce more parameters, and so it is important to control for false positive rates, and there are several publications that have shown that depends on the false positive rates on the number of basis functions in these models, and that it's important to have corrections to the number of effective parameters in the model. And finally, I'd like to just talk about where we can push the limits of PET imaging in terms of timing. And so one of the things that need to be taken into account is the experimental design. Here, we've introduced a method called fPET, where you can use dynamic imaging to look at changes over time in 5 or 10 minute implementation blocks, which is relatively fast for PET imaging. And then this has been taken further to show that even changes down to 1 minute may be discernible using PET imaging. And overall, I like to say that with the advance of new scanners and more sensitive scanners, especially in PET imaging, we may be able to get down to a much higher temporal resolution and try to start to understand molecular processes at a much finer scale. And so I hope I've given you an interesting overview of receptor binding studies with PET and fMRI, and how we can combine both PET and fMRI dynamics, and especially exploit features of time courses in order to understand more interesting molecular processes together with their functional effects. Finally, I'd like to thank everyone that has been involved in these studies and the support from our funding sources.